Good morning, everyone. And we wanted to welcome you to today's webinar. Thanks so much for giving us a couple minutes to get set up here. But my name is Marie Latulippe, and I'm the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IAFNS. And we're really excited to continue our webinar series today. We're actually in about our 7th year of this series focused on all things gut health and gut microbiome. As we start, a bit on IAFNS. And IAFNS is a nonprofit organization focused on advancing food and nutrition science for public benefit. And the way we do this is we bring together scientists from different sectors to identify areas of common interest and then develop projects to advance science in those areas. And as an organization, we do fund research, we convene experts, and we aim to deliver a variety of products. And those products include competitively funded research, workshops, dialogues, and related peer-reviewed publications as some examples. To give you a little introduction to the Nutrition for Gut Health Committee at IAFNS, this slide shows the overall mission of that committee, which is leading the webinar series. And this group focuses on advancing science related to the impact of diet on gut and host health. And you can see here the 4 focus areas, which could be really briefly summarized as looking at definitions and measures, the impact of nutrition on health, mobilizing the evidence, and then we also have a sub-focus on live dietary microbes. All right, as we get into the webinar, just a note on logistics, you should have something like this on your desktop, hopefully, and we welcome you to add your questions in that questions box as things go along. Those will only be visible on the IAFNS side, and what we'll do is we'll use the time we have left at the end of the hour to address some of those questions. And with that, we are quite pleased to jump into the program and introduce Dr. Benoit Chassaing. And he obtained his PhD in microbiology from the University of Clermont-Ferrand in France. After that, he joined Georgia State University to work on some projects related to mucosal immunology and microbiology. And there his work deciphered how genetic and environmental factors, including diet, can perturb the intestinal microbiota, leading to inflammation. And his laboratory is actually now located at Institut Pasteur in Paris. And he focuses on understanding the host microbiota interaction. So today we welcome Dr. Chassaing to share his work on the role played by mucus-associated bacteria in chronic inflammatory diseases, as well as the extent to which microbiota assessment will enable, perhaps a bit in the future, precision nutrition and medicine. So with that, we turn the platform over to you, Dr. Chassaing.
Thanks a lot, Marie. Is it okay? Can you hear me and see me okay and see the screen okay?
Yep, everything looks great.
Okay, thank you so much for this kind introduction and for allowing me to present some of the work that we are currently doing in the lab right now. As you said, I'm going to focus on host microbiota interaction and how we think that studying this interaction can lead to personalized medicine and personalized nutrition. Very likely in the future, of course, we are not yet at this stage, but I really think this is where the field is going. So I do not have any disclosure to disclose related to this current presentation. And so to give a little bit of context and a little bit of background of what we are doing in the lab, we are really interested in the mucosal microbiota, so the microbiota that's closely related and closely associated with the epithelium. And what's quite fascinating to us is if you look actually at the microbiota, it can play very dual role. It can be either beneficial by protecting against pathogenic bacteria and helping us to synthesize, for example, very important vitamins. But at the same time, this microbiota can turn out to be very detrimental and very deleterious for the host, and especially by promoting, for example, chronic intestinal inflammation as well as metabolic deregulation. And it's actually when we were studying this mucosal microbiota, this mucosal niche, and this microbiota associated with the mucosal and the epithelium, that we were able to understand a little bit better what's going on and why in select circumstances, in select conditions, the microbiota can turn out detrimental and move away from its beneficial roles. And this is where we did a good, very important discovery regarding the fact that microbiota encroachment, which is the ability of select microbiota member to colonize the mucus layer and to get in very close contact with the epithelium, that this phenomenon is also is actually very important to drive the detrimental impact of the microbiota and move away from the beneficial one. And I'm going to give you a few examples of how we are studying this microbiota encroachment phenomenon and why we think these bacteria associated with the epithelium, which we call invaders, we think are very important to drive the detrimental consequences of the microbiota. So this is why actually to avoid having these invaders and to really keep the microbiota away from the epithelium, which is labeled here in purple on the bottom of the slide, and you have the bacteria, the microbiota, the bacteria from the microbiota that are labeled here in red. And this is why you have a lot of mechanisms in the intestine that are here actually to help keep those bacteria away and at a very safe distance from the epithelium. For example, you have this very thick mucus layer, which is labeled here in green, that's forming like a gel at the surface of the epithelium, and that's very important to keep those bacteria away. And if you look, for example, at mice that are deficient in this mucus layer, they will develop very, very strong colitis, since they are not able to keep this bacteria, this microbiota away anymore, since they are lacking this protective mucus. If for select reason, as I'm going to give you a few examples during my presentation, you have those bacteria that are able to penetrate this mucus layer and to get in close contact with the epithelium, you will have numerous protective mechanisms, such as all the TLRs and NLRs pathway that will be activated, and that will drive antimicrobial peptides synthesis as well as immune cell recruitment to try to sterilize again this mucosal layer and to try to remove those invaders. So this is another way of looking at it, where you can clearly see on the left side of the screen that this is, under abiotic and symbiotic condition, where you clearly see the bacteria in red that are kept at a very safe distance from the epithelium, while on the right picture, this is one example among many others, where you can clearly see microbiota encroachment, the fact that those bacteria were able to penetrate this mucus layer, get in close contact. And every single time in the lab, we have this phenomenon here as microbiota encroachment. This is always associated with chronic intestinal inflammation. And this is something I'm going to highlight during my presentation. And I will be doing so by taking few examples. So we are studying many phenomena in the laboratory, many actors that can drive microbiota encroachment in both mice model as well as in human. But today I'm going to focus mostly on dietary emulsifiers and how dietary emulsifiers can promote this microbiota encroachment. And I'm going to touch upon also a little bit on prebiotics and how soluble fiber can also modulate this mucus-associated bacterial population in a way that can either promote or demote chronic intestinal inflammation. And again, for both of these examples, I'm going to highlight why we think that studying those bacteria associated with the mucus layer and how microbiota interaction in general will drive in the near future personalized medicine and personalized nutrition. So just to start with the first project I wanted to highlight today, to relate to dietary emulsifiers, which is something we started to study close to 10 years ago now, a little bit more than 10 years ago actually. And dietary emulsifiers are food additives that are highly used by the food industry and that are used to extend shelf life and improve texture of packaged products. And there is a lot of them that are approved to be used by the food industry. But we started our study by looking at two of them, namely carboxymethylcellulose or CMC or polysorbate 80 or P80. And what we did to start with this study was to look at, again, microbiota localization, because we really think that microbiota localization is very important, a very good marker of the intestinal health and host microbiota interaction health in human health. And if you look here at those mice treated with nothing, so those mice are completely controlled and not treated with any type of food additives, you can clearly see that the microbiota is kept away from the surface of the epithelium. And this is actually something we can quantify using confocal microscopy. And we can quantify the distance that separates those bacteria here in red from the surface of the epithelium. And you can see that this average distance is between 25 and 30 micrometers. And we were never able to observe any animals with a distance lower than 10 micrometers. But when we did exactly the same type of measurements on exactly the same type of mice, same strain, same sex, everything was the same, except that this time the mice were treated with CMC or PAT, again, two of the most commonly used dietary emulsifiers. This is what we obtained. We obtained close to a twofold decrease in the distance that separates the bacteria from the surface of the epithelium. And in some animals, as you can see here with a distance of 0, the microbiota was directly touching and adhering on top of the epithelium. And this is, for example, the case here for this specific bacteria, as you can see in the PAT treated mice, where actually some of the bacteria were able to penetrate the full mucus thickness and get in close contact with the epithelium. And again, this is what we think is very central in driving the detrimental impact of dietary emulsifiers on health, as you will see in the next coming slides. So not only dietary emulsifiers impact the microbiota localization and allow the microbiota to colonize and to penetrate this normally sterile mucus layer, but it also drives what we call a much more pro-inflammatory microbiota. So for this, we have some ways in the lab to measure the ability of the microbiota to actually activate and drive inflammation. For example, by quantifying flagellin. So flagellin is one antigen, one of the main antigen expressed by the microbiota that's going to be recognized by TLR5 as well as NLRC4 receptor, and that's going to drive very important pro-inflammatory signaling pathway. And as you can see, if you look in the microbiota from untreated normal mice, you can see that the normal of flagellin that's expressed by the microbiota in those blue bar is relatively stable over the time. But if you start to treat the mice with either CMC or PAT, you can see that now the microbiota is expressing much more flagellin, and you can see this pretty drastic increase in flagellin expression by the microbiota, either treated with CMC or PAT, which we think will be very important in here again driving detrimental consequences, because not only you have a microbiota that's getting closer, but this microbiota it's also getting a little bit crazy and expressing much more flagellin. And again, flagellin is just one example, but we observed other alteration that we think can drive a pro-inflammatory response. So of course, this is what we looked also after in the mice, and for this we used various models, but just to start with the IL-10 deficient mice model. So this is a mice model of colitis, and IL-10 deficient mice will develop naturally colitis. So this is genetical predisposition to colitis, and indeed, if you look at the blue curve, those mice will develop a colitis at approximately 40% of incidence at 3 months of age. But if you treat those mice with either CMC or PAT, now we are doubling the colitis incidence, and we are reaching close to 80%. And not only there is more colitis in those mice, but the colitis we observed were much more severe compared to the colitis observed in the control untreated mice. So this is to us very relevant, because it means that very likely in the human population, and this is something we are currently testing, it means that in the human population predisposed to develop IBD, for example, inflammatory bowel disease, very likely consumption of CMC or PAT by directly impacting the microbiota can very likely drive and participate and promote chronic intestinal inflammation. We did very similar approach on wild type mice model. So we wanted to move away from genetical predisposition and have a look also more to the general population and the general condition without any genetical predisposition. And we observed that in wild type mice, consuming CMC or PAT was also sufficient to drive some level of intestinal inflammation that was sufficient to drive metabolic deregulation. So this is body weight evolution over time. And you can clearly see that after 1 month and a half, 2 months of treatment, the mice treated with CMC or PAT are starting to gain more weight compared to the untreated mice. And this is just one example, but those mice were also developing other parameters of metabolic deregulation. They were developing some parameter of type 2 diabetes, for example, and other metabolic deregulation, such as increase in adiposity deposition and so forth and so on. So it means that in the general population, without genetical predisposition, it's possible that CMC and PAT and very likely other emulsifiers consumption can drive metabolic deregulation, which can play a role in, for example, the epidemic of obesity, as well as the epidemic of diabetes. And in terms of mechanism, and I do not have time to present you all of the data we generated in terms of mechanism, but we were able to demonstrate that the microbiota is actually the direct target of those compounds, and the microbiota is going to detect those dietary emulsifiers and respond by colonizing the mucus layer, expressing much more flagellin in a way that's going to drive chronic intestinal inflammation. And this inflammation can manifest as colitis, as I showed you, if there is also associated genetical predisposition, or if there is no genetic predisposition to metabolic deregulation. And so what's also very interesting to us, of course, is the inter-individual variation. And just to finish with the mice data, and before moving to the human research clinical trial that we recently performed, is when we looked at the mice data, we were very impressed and very intrigued to observe that actually not all the microbiota are responding the same to dietary emulsifiers. And we were able to observe that there is some mice that harbor a resistant microbiota that even if you give them dietary emulsifiers, they will not actually respond to dietary emulsifiers, and they will be fully protected in light of the resistant microbiota that they are hosting in their intestine. On the other hand, we observed some mice that were highly sensitive and highly susceptible to the dietary emulsifiers, with the microbiota hosting select species that, when exposed to dietary emulsifiers, can drive chronic intestinal inflammation and metabolic deregulation. And we are doing a lot of experiments right now to identify those bacteria that can drive sensitivity to dietary emulsifiers in both mice as well as in human, as I'm going to present you in the next coming slides. So, of course, then it's coming the question of the human relevance and what's the human relevance of these findings. So there is definitely some human relevance, especially regarding epidemiological data. So there is this very important French cohort on epidemiological association between dietary habits and health, led by Mathilde Touvier, which is named the Nutrinet Santé cohort. And they were able to observe in this cohort that there is a positive association between the consumption of select dietary emulsifiers and the risk of cardiovascular disease, the risk of type 2 diabetes, as well as the risk of various cancer. So, of course, this is epidemiological observation. So it means that there is a link. It means that there is a correlation, but it doesn't necessarily mean that one is driving the other. So this is something we are currently working on. So this is the first research clinical trial that we did a couple of years ago, and right now we have a much more ambitious clinical trial. We can touch upon it during the Q&A session. But in this first ever clinical trial on dietary emulsifiers in human, what we were able to do was to perform. So this is really pilot clinical trial, because we were focusing only on one dietary emulsifiers, which is carboxymethylcellulose, only for a really short period of time. So we were able to follow those patients for only 14 days and on a very limited number of participants. So we had a total of 16 participants, which is again very pilot and very preliminary. But what we did nonetheless was, I think, very important, because what we did was we were able to have those patients locked in an hospital room, because we wanted to be really sure about what they were eating and what they were drinking. And we also wanted to have access to their fecal samples on a daily basis. And half of those patients followed the additive-free diet, so they consumed a diet that was not containing any type of food additives, while the other half of the patients were consuming a diet that was supplemented with dietary emulsifiers, carboxymethylcellulose. And again, we were collecting feces on a daily basis, and we were also able to collect biopsies on those patients just before exposure to CMC, as well as just after the end of the exposure to carboxymethylcellulose. And just to present briefly the data on the microbiota composition, we were able to observe that just consuming CMC for such short period of time was nonetheless sufficient to drive alteration in microbiota composition, as you can clearly see here with the differential clustering between CMC exposed participants and non-CMC exposed participants. And looking at alpha diversity also, which is a very good marker of microbiota health. So the higher the alpha diversity will be, the more beneficial the microbiota will be. You can see that in the participants that were not treated, the alpha diversity was staying relatively stable, while as soon as we expose the participants to CMC, carboxymethylcellulose, you can see this slight drop in alpha diversity of their intestinal microbiota. So again, this is exposing the participants only with one dietary emulsifiers on a relatively short period of time, but nonetheless, this is sufficient to drive a short depletion in microbiota richness. So what will be the consequence of years and years of exposure to a cocktail of food additives? This is something we are currently investigating. And again, as I mentioned, we were able to collect from those patients biopsies. So if I move back to this slide here, we were able to collect biopsies just before exposure to CMC and at the end to the exposure to at the end of exposure to carboxymethylcellulose. And we looked at microbiota localization because again, we think that this is a very important marker of host microbiota interaction health. And what we observed is if you look at the control patient, and this is a log2 scale, so meaning we compare post-exposure compared to pre-exposure. So if it's at 0, it means that the distance was not impacted. If it's above 0, it means that the distance was increased. If it's below 0, it means that the distance was decreased after exposure compared to before exposure. And if you look at the control group, you have a distance of 0, which makes perfect sense because those participants were not treated with anything. So basically, the microbiota localization stays the same after compared to before, which makes perfect sense. But if you look at CMC treated participants, we were actually at the beginning quite disappointed by this observation, but there is clearly 2 subgroups of participants. 5 out of the 7 seems to be fully protected and not at all impacted by CMC exposition. So you can clearly see that the microbiota localization is staying the same or maybe even slightly increased in those participants after exposure to CMC compared to before exposure to CMC. While, on the other hand, for 2 out of the 7 participants, there was a very strong microbiota encroachment that was promoted in those participants by being exposed to CMC. So we were quite disappointed by this observation, but nonetheless, this was again bringing back the concept that very likely not all the microbiota are going to respond the same to dietary emulsifiers, and that there is very likely some microbiota and some people will host some microbiota that will very likely make them much more sensitive and much more susceptible to dietary emulsifiers, while some people may host microbiota that are highly resistant to carboxymethylcellulose. So this is something that Noemi wanted to test when she was a postdoc in the lab. And what she did actually was, I think, pretty elegant. And she performed fecal microbiota transplantation to germ-free IL-10 deficient mice. So we took mice susceptible to develop colitis because we wanted to have inflammatory readout in these experiments. And she took germ-free because we wanted to have mice without any microbiota. And we used those germ-free mice as a recipient for fecal microbiota transplantation, coming from either 2 out of the 5 microbiota patients that were identified as CMC resistant, which are the blue donor, or 2 of the patients that were identified as being highly sensitive to carboxymethylcellulose. So she performed those fecal microbiota transplantations. So she ended up with 4 different microbiota, 2 resistant and 2 sensitive. And then we allowed the mice to stabilize the microbiota for 2 weeks. And then we subjected them to either water or CMC exposure. And what she observed, I think, was really, really interesting and really promising is if you just look at histological scoring, for example, on the right slide, so colon length is also a very good marker of inflammation, but just looking at histological scoring, you can clearly see that when Noemi colonized mice with a microbiota that were identified in clinic as being CMC resistant, and when she gave CMC to those mice, they did not develop any type of colonic inflammation. So it seems that those mice were actually fully protected against the promotion of inflammation induced by CMC. But when she did exactly the same, but this time in mice colonized with CMC sensitive microbiota, this is what she observed. She observed that the mice colonized with CMC sensitive microbiota developed very strong colitis when they were treated with carboxymethylcellulose. So I think it's a very important concept here because it means that very likely CMC by itself is not sufficient to drive colitis because if you have a resistant microbiota, you will be fully protected. On the other hand, if you have a sensitive microbiota but you are not consuming CMC, this is not sufficient to drive colitis. You see, those mice here are not developing colitis. But if you have the combination of the sensitive ecosystem plus exposure to carboxymethylcellulose, now you have the combination, and then those mice are developing very strong colitis when they are subjected to CMC and hosting a sensitive microbiota. very similar observations were made when we looked at microbiota localization, which again we think is a very good marker of intestinal health, where if you look at mice colonized with CMC resistant microbiota, there is some level of microbiota encroachment that were induced by CMC exposure, but this was non-significant and to a low extent. But when we did the same measurement on mice colonized with CMC sensitive microbiota, this is what you got with this very strong microbiota encroachment, which again makes sense because it was associated with colitis, but only in mice colonized with CMC sensitive microbiota. So again, this is driving the concept that very likely there is some microbiota that will host select species, and we are currently working on investigating them, that will drive colitis when they will be exposed to CMC. And on the other hand, you can have some microbiota that will be fully resistant to dietary emulsifiers, either because you do not have the right trigger to drive inflammation, or because you are also hosting bacteria that can protect you against the detrimental impact of dietary emulsifiers, such as Akkermansia muciniphila, as we very recently report. So we did some, as you can see, everything started more than 10 years ago in mice, and then we did the first human clinical trial, and then we moved back to mice to understand the mechanism and to understand how the microbiota can drive a personalized response. And we are also working more and more with in vitro microbiota system, which is what's presented here, and this allows us to cultivate in parallel up to 50 microbiota. And this is a very elegant tool because when Chloe, Erika, and Eloise in the lab use this type of in vitro microbiota system, what they observed is actually you can reproduce in this in vitro microbiota system what we observed before in human or in mice. Meaning that if you colonize the system with microbiota that's fully resistant, so coming from the resistant individual observed in clinic, and you treat those microbiota with CMC, PAT, or a combination of both, you can see that the microbiota is actually not at all responding in, for example, its flagelling level, which is a very good marker of microbiota response to dysbiosis and to perturbation. But if you do exactly the same, this time in vitro microbiota system that were colonized with microbiota coming from a sensitive individual, this is what they got. They got a very strong and synergistic response when you expose the microbiota with either CMC or combination of CMC plus PAT. So meaning that, again, what we think will be really cool, and this is something we're currently investigating, is to use this type of in vitro microbiota system to investigate microbiota sensitivity status to dietary emulsifiers and see if we can actually predict emulsifier sensitivity to microbiota sensitivity to dietary emulsifiers. And just and again, we are currently doing it with a much larger clinical trial that's enrolling close to 200 IBD patients, but again, we can discuss a little bit about that during the Q&A session. And just to discuss a little bit about prebiotics, so as I mentioned, I wanted to discuss a little bit about what we did on dietary emulsifiers, but we are also doing more and more work on prebiotics and soluble fiber, and how we think the response to soluble fiber is also going to be highly personalized. So here, this project started with Erika, that wanted to look at the effect of various prebiotics in our in vitro microbiota system. So what she did was to inoculate this in vitro microbiota system with the microbiota from a healthy donor, and looked at the response of those microbiota to soluble fiber inulin, soluble fiber xylem, or as a control to relatively insoluble fiber cellulose. And what she did, and what's the beauty of this in vitro microbiota system is, as I mentioned, we can have up to 50 microbiota in parallel. So of course she did not do it on one donor, but she performed this analysis on 6 different healthy donors, meaning that we can investigate the entire individual response and the personalized response to dietary emulsifiers. And again, cellulose was used as a control, and we looked at the response of the microbiota to inulin and xylem, which are 2 commonly used soluble fiber. And what she observed, so I'm not going to go into all of the detail because it's a lot of analysis and it's a lot of sample processing, but because what she did was to look longitudinally on those microbiota and to look for microbiota composition, microbiota function, generation of short-chain fatty acid, microbiota pro-inflammatory potential. So she performed really an in-depth characterization of those microbiota. And after, what she did was to perform a multivariable analysis, so meaning we combine all of those measurements together to look at the response of the microbiota. And this is exactly here what the Breker's distance is doing. So basically, we are combining all of those variables and looking at how the microbiota responds to dietary emulsifiers, to soluble fiber, sorry. And if you look for this first donor in blue, you can clearly see that if you treat the microbiota with cellulose, inulin in the middle, or xylem on the right, you do not have much of a response compared to the cellulose treatment. So you see inulin and xylem are not driving any type of response, any type of microbiota modulation compared to cellulose exposure on this specific microbiota. Meaning, and again, we use relatively low dose of soluble fiber. So meaning that for this specific donor, exposition to inulin or xylem was not impacting microbiota composition or microbiota function at all. But when she did the same type of experiment and the same type of analysis for all of the donors, she clearly observed, again, a bimodal distribution where, again, donor 1, donor 5, and donor 6 in purple and gray, respectively, were not responding to inulin or xylem, while for some other donor, and this is especially the case for donor 2 in red and donor 3 in orange, you can clearly see that if you treat the microbiota with either inulin or xylem, you have this very strong response of the microbiota. And this is true for composition, so you have alteration in microbiota composition, but also for microbiota function, you have alteration in metabolite production, you have alteration in the pro-inflammatory potential, and you can see very, very strong response induced by inulin and xylem in both of these donor 2 and donor 3. So this was, again, bringing the concept that very likely not all the microbiota are equal in facing soluble fiber, inulin and xylem. And this is, identification of microbiota that seems to be highly resistant to fiber, and the identification of microbiota that seems to be highly sensitive to soluble fiber-induced modulation and induced modification. So this is, again, another way of looking at it, but what she wanted to do next was to validate it in mice model. So what she did was very similarly to what I presented you before for soluble fiber, was to colonize a group of mice with a microbiota that was identified as being highly resistant to fiber, or with one microbiota that was identified as being highly sensitive to fiber. And again, we performed microbiota stabilization for 2 weeks, and then we looked at the colitis sensitivity in those mice after treating them with either cellulose, inulin, or xylem. And what she observed, I think, was, again, really interesting and really promising. So again, if you look at histological scoring, you can see that in the mice that were colonized with a resistant microbiota, so this is donor 1 here. So you see a microbiota that do not respond at all to inulin or xylem. And you colonize the mice with this microbiota, you treat the mice with either inulin or xylem, you can see that the DSS-induced colitis is not at all modulated, not at all impacted by either inulin or xylem. And this is also the case when you quantify CD68 positive cells, which is a very good surrogate of intestinal inflammation in this DSS-induced colitis. You can clearly see that in those mice, the level of inflammation is very similar whatever they are eating, either cellulose, inulin, or xylem. So there is no fiber-induced modulation of the colitis. But when she did exactly the same type of experiment and exactly the same type of measurement in the mice that were colonized with a sensitive microbiota, so again, if I go back with this microbiota 2 here, that seems to be highly modulated by inulin or xylem, this is what she got in the mice model. She got mice that were highly impacted by either inulin. So we observed that inulin was actually promoting colitis and really, really very severely increasing the severity of the colitis, while xylem was actually protecting against DSS-induced colitis. So this kind of makes sense because there was also previous publication, including by us, showing that inulin can actually promote inflammation, while xylem can actually protect against inflammation. But what's clearly new here in this data is that this seems to be highly microbiota dependent and very personalized, meaning that if you have a microbiota that's highly resistant to fiber, you will not be impacted by inulin or xylem, while if you have a microbiota that's highly sensitive to soluble fiber, then you are going to drive a colitis that will be highly modulated by either inulin or xylem, with inulin promoting inflammation and xylem demoting inflammation. And just to finish very quickly, because I'm running out of time, but we also have a very interesting project on probiotics, and especially Akkermansia mucinifila, which is a very fascinating microbiota member. So this bacteria can actually found in many people, and it's supposed and it's suspected to be able to beneficially modulate host microbiota interaction and to drive beneficial metabolic benefit to people, and there is a clinical trial using this bacteria. So it's kind of a fascinating bacteria and a fascinating probiotics. And what we wanted to investigate here was very similar, was to look if maybe Akkermansia mucinifila can also actually directly modulate the microbiota directly in an host-independent manner. So for this, we use the in vitro microbiota system, and if this can be donor-dependent. So I'm going to go very quickly through the data, but basically what we did was very similar to what we used on prebiotic, was to use our in vitro microbiota system to inoculate the system with 12 different healthy individuals, and to look longitudinally for the effect of Akkermansia mucinifila in modulating the microbiota compared to PBS, control untreated chambers. And I'm going to go through this data, but what we observe here is very similar to what I presented you before, is when we combine all of the data in terms of microbiota composition and microbiota function modulation induced by Akkermansia, we observe that most of the microbiota are actually not responding to Akkermansia mucinifila, while some of the microbiota were highly beneficially impacted by Akkermansia. And you can clearly see that those 4 microbiota were responding quite dramatically to the presence of Akkermansia when we treated and when we exposed those microbiota to Akkermansia in a way that were highly beneficially modulated. And what Clara did so again, this is this concept of resistant donor to Akkermansia as well as sensitive donors to Akkermansia. And just to finish on this project, we did very similar approach of microbiota transplantation. And what she did was, as I presented you before, was to select one microbiota that seems to be highly sensitive to Akkermansia and one microbiota that seems to be highly resistant to Akkermansia. And with Clara, we've transplanted those microbiota to germ-free mice. And after transplantation, we looked at the beneficial modulation induced by Akkermansia mucinifila in high-fat diet-induced obesity. And what we observed, I think, was, again, really striking, and we are going to publish this data really soon, hopefully, was in mice colonized with a microbiota that's fully resistant to Akkermansia mucinifila. We observe that Akkermansia mucinifila in those mice is not driving at all any type of metabolic improvement. But in mice colonized with microbiota that's highly sensitive to Akkermansia mucinifila, we were able to reproduce what was observed before many times, which is a beneficial metabolic benefit induced by Akkermansia mucinifila. So again, there is nothing new here in observing the metabolic benefit of Akkermansia mucinifila, but what's very intriguing and what's very new is that this seems to be microbiota dependent and very personalized, and very likely Akkermansia mucinifila is not going to provide metabolic benefit to the general population, because you need to have a good microbiota and a sensitive microbiota to drive the metabolic benefit induced by Akkermansia mucinifila. So just to finish and to conclude before answering the question, again, what I wanted to demonstrate and what I wanted to highlight using dietary emulsifiers, prebiotic and probiotic, is for all of these compounds that interact and can modulate our microbiota, we think that this is highly personalized, and there is definitely some microbiota, some individuals that will be highly resistant, and some individuals that will be highly sensitive to the perturbation induced by emulsifiers, prebiotic or probiotics. And we think that here, in this resistant versus sensitive dilemma, we think that the microbiota is actually a central player, a central nexus in driving such personalized response to diet, prebiotic and probiotic. And what we think will be the future, and we are currently doing a clinical trial for these aspects, is actually use some of this data to be able to stratify the patient in order to perform microbiota-based precision nutrition and precision care by using microbiota data to look at the most beneficial dietary recommendation and the most beneficial care in terms of, for example, preventing IBD or metabolic deregulation. And if you look in the past, in the last 20 years, there was a lot of study looking at and comparing the microbiota between healthy population and diseased population. But if you look what the field is moving now, is really more looking at this inter-individual variation and looking within, for example, the IBD population, how we can, within this IBD population, use the microbiota data to have nutrition, personalized nutrition and personalized medicine based on the microbiota. And this is exactly what we are currently doing in the laboratory. So of course, this is a team effort. So I wanted to thank all of the people involved in driving and in helping and running all of these experiments that I was presenting you today. This is also, of course, highly collaborative. So we have many collaborators all around the world that's helping us in setting and doing these experiments. We have a lot of funders also to support our work. So I wanted to thank them one more for supporting our work and research. And yes, I will be really, really happy to answer any question you might have. Thank you so much.